All right, so centrioles in normally prophase or early prophase, the first thing they do since they're pairs is they start to separate. All right, so they start to begin to go towards opposite poles of the cell. That's the first thing centrioles start to do. All right, so after prophase, we have metaphase. This is where the chromosomes start to line up in the middle of the cell. By then, the centrioles would have already started to develop a thin-like thread, all right, which you would call spindle fibers, all right? You know, the things that look like hairs that come out of it. Those spindle fibers will eventually start helping move these chromosomes, all right, to separate parts of the cell. That's one part of what centrioles do, is create the spindle fibers to help move the cells, to help move the chromosomes around in the cell. All right, so now in anaphase, the centrioles will use the spindle fibers to separate the chromosomes into individual chromatids. All right, so each spindle fiber, and since there's centrioles on both sides, will pull apart the chromosomes to make them chromatids, one on one side, one the other. Spindle fibers separating them. All right, so that's what the main simple basic thing centrioles do in the anaphase. So after the chromosomes finish moving through the microtubulars to the opposite ends of the cell, they disappear in the telophase. So you no longer see the thread-like fibers that move the chromatids to opposite sides. So now their job is done. So the microtubulars disperse, and then the division of the cell membrane begins now. And that is the job of the centrioles so far in mitosis. Alright, so what are centrioles? You'll find them in animal-like cells. They come in pairs, and they're very important for the cell division and mitosis. So let's get into what centrioles do in each phase of mitosis. Alright, so mitochondria. You'll find it in plants, you'll find it in animals, and you'll find it in human beings. So there are these things called a cristae. You'll find them in the folds around the matrix in the mitochondria, and they contain this protein that helps the reaction that causes ATP. Now, ATP is what gives the cell energy to do the cell division, and the cells need energy, so we can fuel our bodies with that. Because of the way mitochondria is shaped, it gives it more space to create more energy. Okay. Not only that, mitochondria has their own DNA, and it's different from all the other cells that we have in our body. Okay, so that's one unique feature about it. Because a lot goes on with mitochondria and how it's consistently doing chemical reactions, you have different levels of membranes. You have the outer membrane, and you have the inner membrane, and you have the matrix. Alright, so it's one of the most important features of our body, mainly because of the ATP, which gives energies to our cells. Golgi apparatus. So it's a series of membranes that are shaped like pancakes, almost. That's a good resemblance. And so they're responsible for, it's almost kind of like a UPS store. It'll gather simple molecules and combine them to make, bigger and more complex molecules. And then once they get these molecules, they'll start to package them in these things called vesices, and it'll either store them for later or send them out to parts of the cell that needs it. And then the Golgi apparatus is also responsible. They build lysosomes. These lysosomes, they're basically the cell digestion machine. So that's an important factor of the Golgi apparatus. Easy ways to think about it is like UPS stores. Packs, modifies, then ships it out to which part of the cell that needs it, or it could just, hold it in its warehouse for later. So since we talked about the Golgi apparatus, let's talk to another organelle that works very well with it, and that's the rough and smooth ER. So here are the pancakes, like the Golgi apparatus. Here are some of the vesices and the lysosomes we talked about. Smooth ER, rough ER. So, the smooth and rough ER, they both have the same type of membrane, but they do have different shapes. The rough ER is a little bit more bumpier because they contain ribosomes in there, while the smooth ER is like the name, is more looks like tubes. All right, and of course they're smooth because it doesn't have the ribosomes attached to it. The smooth ER is more of a kind of a storage organelle. All right, it keeps lipids and steroids in there, that's what you'll find in there. Okay, and then the rough ER, it does have the ribosomes, and ribosomes are an important part of proteins, packages and synthesize the proteins. All right,